with Dr. Mike Lomas, Director of the National Center for Marine Algae and Microbiota and Director of Bigelow Center for Algal Innovation. So we will introduce his different seaweeds collection. Mike, over to you.
Wonderful. Thank you, Melanie. Thanks, everyone. Yeah, so over the next few minutes, I'm going to talk about, the collections we hold here, just so to bring everyone up to speed, because some of you may not know what NCMA, or the National Center for Marine Algae and Microbiota, is. It's a U.S. Algal germplasm. It's been around for 43 years, plus or minus. We serve a public mandate of being the home for wayward algal souls. So I forget which earlier talk this morning said that, as funding runs out for other collections, private research collections and things like that, there's a facility that actually will bring them in and still make those research assets available. And so that's the primary mission that we hold. And so over the course of the last 40 years, we've been growing our algal germplasm collection by our own collections, as well as bringing in these sort of orphan collections. And so we have roughly 2,500 microalgal species in our collection. But today I'm just going to talk about the macroalgal side of the collection, which is about 1,400 strains, plus or minus. So next slide, please. Excellent. Thank you. So within the NCMA collection, there's this general algal collection. These are sort of the accumulation of those orphaned collections. And so this is a lot of work from John West and other researchers like that. I've got listed here the genera and the species, but there are 20 or so genera, 30 or so unique species, 146 physically distinct isolates, all collected from around the world, but primarily the tropical domains, a few from Northern Europe. Many of them are cryopreserved. Some we keep in perpetual culture, just because we haven't gotten to the actual cryopreservation techniques for them yet, as we sort of work through the collection. You can go ahead to the next slide, please, Melanie. Thanks. So there's also within the NCMA macroalgal collection 2 sub-collections. And so one of them we call this Yukon NCMA collection. This is Charlie Yarish's research collection. When he retired a couple of years ago, he transferred those holdings to NCMA to make sure that those assets continued to be available to the research community. And so here's just a quick description of that sub-collection. It's about 8 genera, 20 species, 250 or so isolates, again, from around the world, but now focused more heavily on sort of the northern subtropics and towards the northern subpolar domains. You can go to the next slide, please, Melanie. In addition, there's also another sub-collection that's called the Hui Yukon NCMA sugar kelp sub-collection. And this is a sub-collection created by Scott Lindell, who's on the webinar or the conference this morning, and Charlie Yarish, where they focused on one species, Saccharina latissima, and collected a lot of individual gametophytes, both male and female, to try to get an idea of that biodiversity within that one genus and species. And so just to point out that a lot of that phenotypic and genetic characterization data is available on the sugarkelpbase.org data website. Go to the next one, please, Melanie. Wonderful. So, there we go. So I just want to take the rest of my time to take and talk about some of the things that we do within NCMA to try to tackle some of the challenges that have already been brought up today, like security, data, things of that nature. So within our collection, we have a number of very detailed security protocols. Obviously, the first one is routinely keeping an eye on the organisms themselves, on the macroalgae themselves. So there's a lot of hand and eyeball time on them to take and be mindful of potential pests or other invasives, or sometimes microalgae get in there or grow up under particular conditions. Within all of our holdings, we have multiple redundant incubators at each temperatures, so that if one were to fail, mechanically fail, we can move things around so that we don't lose strains. We have a number of different monitoring systems, one internal and one external to the physical laboratory itself, that when there's a problem, when there's a failure of an incubator, whoever happens to be on call that day gets a call and says there's a problem, run to the lab and move things around. And for some of our more critical or more valuable strains, we actually work with the U.S. Department of Agriculture, Agricultural Research Service, as a backup physical off-site storage location of some of those important species. And we try as best we can to use cryopreservation as a more viable management strategy, right? We accept the fact that there's never going to be enough money to actually maintain these correctly. So if we can cryopreserve them, that's a way to use it as a management tool to minimize the actual people hours needed for that management. Can you go to the next slide, please? There we go. One of the things that we are working through is how do we try to aggregate the data to ensure that it's actually accessible by the research community. There's, for the Yukon Hui collections, a lot of that data exists within, as I said, the sugarkelpbase.org, where they have on our website, we have a lot of the metadata with those strains, cryopreservation information, and some other parts of information. So even within those, the knowledge about them is split over two places. Within our macroalgae collection, we're trying to work towards building out some base level information, whether it be growth rates, whether it be nutrient contents, or high biochemical class like protein, carbohydrate, lipids contributions, as a baseline level to ensure usefulness, right? When people want to think about what they might use, what they might keep or not keep, we need at least some information to inform those decisions. And so that's our goal here, is to try to create ultimately sort of this baseline knowledge. And then hopefully we can ingest other data to improve the overall knowledge value. You can go on to the next slide, please, Melanie. This is my common lobbying on data, is that because of the way the U.S. funding system works, every agency, it seems, to have its own place it wants data to go, and therefore it results in a very fractured data system within the U.S. And so if I want genetics data, I go here. If I want phenotype data, I go there. And it makes it really, really hard to actually then ingest, because they all use different vocabularies and ontologies and et cetera, et cetera. So one of the goals that we are trying to lead within the U.S., and it was great to hear that first thing in the C strains talk and the earlier talk, and we should definitely talk, is how do we take and start to use similar vocabulary, similar ontologies, so that actually databases are interoperable and therefore searchable by anyone globally. We talk about global data and knowledge. How do we actually make it globally accessible? You could go on to the next slide, please. Another important thing to think about, for us anyway, because we are a germplasm depository, we distribute these algae globally to both academics and companies. And so IP ownership and access to the physical samples is really, really important for us. And so, we use material transfer agreements when we work with companies. We don't when we work with academics. They generally have different goals and what they're using it for. We do strain licensing. At least in the U.S., there's this belief that I believe in, that companies should actually help support public assets. They're making money. They should not make money off the back of academics. So how do we figure out access, but valued access? And so thinking about IP structure and things like that is really important. If you could go to the next one, please, Melanie. And so also affiliated with this is now how we more effectively work with our national funding agencies, and this might be relevant to Europe funding agencies. In the US, our National Science Foundation is now working through what is called a sample management plan, whereby germplasm collections such as ourselves, if we are expected to hold material that is isolated as part of a federally funded project, we can get access to financial dollars through that grant to actually help maintain it. So we're starting to get better connection between the collection of the resources as well as the support to maintain it. And I just throw in these last two bullets below because they're things that I'm involved with directly. The USDA's, U.S. Department of Agriculture's National Genetic Resource Advisory Council is really pushing hard on how to integrate algae into the concept of a management system, much like our national plant germ system or the crop diversity trust that we heard about earlier. So out in the US anyway, algae is getting a bigger seat at the table. Now we just got to figure out how to better integrate them by talking the same language.